Okay, so in this video we are going to discuss about control of development. This is quite a small topic. Like this topic is simply controlling development by we have two factors. One is nucleus, controlling role of nucleus in development, and second is role of cytoplasm in the development. So let's check it out. So first thing, control of development. It's simply we know how fertilization take place when ovum and sperm get fertilized. So if this the ovum and sperm is here, so it's called as fertilization, right? Fertilization. So after this fertilization they will form fertilized egg, and that's also called as zygote. So zygote is basically single cell organism. So for role of development or mechanism of development is simply when the single cell get transferred to multicellular complete organism, that's called as development. Basically that's called as these all things like multicellular, which mean how a single cell from fertilizing ovum and sperm we get a new child, new baby born children, right? This means they're turning from from single unicellular to multicellular complete organism that would have of course tissue, organ, even the organ system. So you see, like everything is quite natural, and we are so blessed with this all. So this is quite a small topic. I hope let's say you like get to know more things over here when we read this. So let's start. The control of development, which involve interactive interplay between genetic and environment factor, which determine how a fertilized egg, which is simply fertilized egg and zygote, both are the same thing. They transform into complex multicellular organism. This means complex, which means quite difficult. As we know, a human being is quite complex structure just because it has so many with distinct tissues, organs, and all. So key process is the control, which includes pattern formation, the different regions of the embryo, different specialization through gene expression, and this gene expression is also quite important. Either that's in signaling pathways. Another essential aspect is cell differentiation, undifferentiation cells, and these all things factor. Even molecules or additionally the environment including factors like temperature, light, nutrients also influence gene expression. Even they also control gene expression, either the temperature, light, and nutrients. And cell differentiation thereby impact the overall development of the organism. So it's quite a small topic. So now we are going to discuss role of nucleus in the development. Role of nucleus in development. I'm not writing in development. When it comes role of nucleus, first thing, they help in gene control, right? Gene control. So this gene expression, which is produced by nucleus, basically produced by nucleus, and when we talk about the found, where do we find this? We found this in a cytoplasm. Okay, so then comes another part, which is in this one we have taken an example that's called as acetylbluria. Basically this is a multicellular alga. That's basically multicellular alga, green alga. And then this has so many, there 10 size or the so many things. Let's discuss it out. So here comes the structure. So let's make them. Here comes the first part, then here we have the line like this, and so this is the basically structure what you'll see. Okay, so the first part, this is called as cap. When we talk about the cap, this means it's an umbrella like structure. right? Umbrella like structure which is present at the top. Of course we can say it's present on the top. Then this cap is like simple regular shape, it should always be round. Then comes the second part like this one. This the middle part here called as stalk, which means it's a long stalk which arises from the rhizoid. Arise from the rhizoids. Then the last part comes, this one is called as rhizoids. We simply attach to some substance, and basically this have single nucleus. So I'm sure it will be clear. So there was an experiment done by this experiment done by Hamling Hammerling. So what he introduced two kind of divisions or what. So first thing and the second one, first one was a Mediterranean. So that's basically a Mediterranean. And the second part which comes called as a crenulate. So what are these? And this means we have taken two examples. So that examples are quite easy. It's simply if we just take the cap which is regular shape, as I mentioned before, regular shape which means okay and perfect, which have a structure like this, right? So this is perfect, right? Then in this a crenulate we just took this kind of shape which show that this is not regular shape, this cap is irregular. So what will happen then, this is the first part, then comes the second part. In that what we does, here was the same part till now here. Then what we are going to do, okay, here we are going to add the second part of the a crenulate, and then again the same part like this, and here comes again this shape. So here's the second part as well. So this means we have changed, he cuts not he, but of course we cut off nucleus. So here we just cut off nucleus. If I just write it, cut off nucleus. When we just cut off nucleus, this containing rhizomes from an algae which is a differentia, which is the first example. And then we just took from a differentia and we have grafted, which means not uploaded, but we have embedded into another piece of alga. That's basically called that a crenulate. So in the result there was no change. This means nucleus really matters. So when comes the result, so we are mentioning, so when comes result, nucleus contain all genetic information. This was proof. The structure does not matter, but the nucleus contain all genetic information. Which is essential for development. Right? And cytoplasm also play good role in selection of gene. This could be also a good point. Cytoplasm play role in selection of gene. So yes, here comes the end. But before we end, let's see what's written in a book. So let me add this picture again. I know these are blurred quite. So here comes role of nucleus in the development. During his studies in 1920, the experiment was done by Hamring, which he discovered acetobularia, which is type of marine alga, multicellular marine alga, which has a single nucleus located in the base of the cell. and conducted this experiment in 1934, successfully transplanted nucleus of one acid bacteria species to acid bacteria crenulata on stem of another species. This means they are adding stem of another right here. Then acetic bacteria, which is Mediterranean, not Mediterranean, but this is Mediterranea, which is the second type, which had a different cap shape. So cap shape was different in this one. So the interesting finding was that the cape grew in the transplanted cell resembled that of A. So this means cape was growing with the resembling of the shape of A crenulata. This indicates that the nucleus influenced the development of the cap. This means nucleus was influenced by the development of cap by transmitting factors substance to the cytoplasm. So this means no matter if you change the shape and at all, still the transmitting factors substance to the cytoplasm in simpler terms are quite good so that it could be transferred. The experiment showed that the nucleus controls the information and determine the shape of cap, which I already told you. So I'm sure it will be clear. Then we'll come with another video which would be about development or the role of cytoplasm in the development. So yes, thank you for watching. Bye bye.